In this video, we will be going through enzymes, which is a part of the biological molecules topic. If you haven't seen the carbohydrates, lipids or proteins video, please head over to my channel and give them a watch. Enzymes are biological catalysts which increase the rate of biochemical reactions without being used up. Enzymes are a type of protein which has a tertiary structure which determines the shape of the active site. Enzymes can be either intracellular, which are produced and work within the cell. The other is extracellular enzymes, which are secreted and catalyze reactions outside the cells. Enzymes have an active site which is specific to the substrate. Once the enzyme and substrate have bound, it forms temporary bonds and are called enzyme-substrate complexes. The enzymes can catalyze catabolic reactions which break down big molecules into smaller ones, or anabolic reactions which build bigger molecules. However, remember this is that a catapult breaks things, so catabolic equals breaks down. Once the enzymes have catalyzed the reaction, the products are released. In your exam, anytime there is a question talking about enzymes, make sure to try and use the terms active site, substrate, enzyme-substrate complexes and complementary. There are two models that you need to be aware of for how an enzyme fits with a substrate. There is the old lock and key model and the current induced fit model. The lock and key model simply suggested the enzyme acting as the lock would fit to a specific key, which is the substrate, and suggested they were rigid structures. Although this is partially true, the induced fit model evolved to suggest when the substrate reaches the active site, conformational change occurs, allowing the active site to mold around the substrate. Enzymes are catalysts, which mean they lower the activation energy needed by providing an alternative pathway. Activation energy is the amount of energy needed for a reaction to take place. Therefore, enzymes lower the amount of energy needed for substrates to become unstable in order to form the products. This diagram shows you the substrate, the products, and the activation energy needed, both with and without a catalyst. There are four main limiting factors that you need to be aware of with enzymes. Enzymes are proteins, so anything that can denature a protein can denature an enzyme. If an enzyme is denatured, the active site will change shape and no longer be complementary to the substrate. Therefore, less enzyme-substrate complexes can form, so less products will form, leading to reduced rate of reaction. The first limiting factor that you need to be aware of is temperature. At low temperatures, the substrate has less kinetic energy, hence will move slowly and less frequent successful collisions will occur. A successful reaction is one where the reaction is complete and ends with products being formed. Therefore, less enzyme-substrate complexes will form. Whereas at higher temperatures, due to the increased kinetic energy, more frequent successful collisions occur, maximizing the rate of reaction. However, if the temperature is too high, bonds in the tertiary structure will break and cause the enzyme to denature. As the enzyme active site has changed, no enzyme-substrate complexes can form, stopping the reaction. The next one you need to be aware of is pH. Enzymes work within a narrow pH range, as out of this range the hydrogen bonds and ionic bonds in the tertiary structure will be disrupted. Then we have the enzyme concentration. As the enzyme concentration increases, the number of active sites that are available increases, so there's an increased chance for enzyme-substrate complexes to form. However, a plateau is reached when the enzyme concentration is no longer the limiting factor. Finally, substrate concentration. As the substrate concentration increases, the rate of reaction increases, but it plateaus as the enzymes are working at their maximum capacity, also known as Vmax. No more active sites are available, therefore substrate concentration is no longer the limiting factor. In your exam, if there is a question about temperature as a limiting factor, remember to talk about kinetic energy and frequency of successful collisions. If they mention pH, remember to talk about ionic and hydrogen bonds. If they give you a graph and ask you to calculate the rate of reaction, you need to draw a tangent and calculate the gradient. So, the gradient is the change in the y-axis over the change in the x-axis. If it is the initial rate of reaction, just bear in mind that that means the rate of reaction at the start. So you need to draw a tangent from the start of the graph where the time is 0. I hope you found this video useful. I will be going over enzyme inhibitors in the next video, so be sure to give that a watch too. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.